Hi there, and welcome to the Chapter 1 lecture for microbiology. The first chapter is it's all introductory, right? So we're going to talk an overview of different types of microbes and some of the basic chemistry and cell biology involved in studying microbes. So microbes, by definition, it's really a short term or a slang term for microorganisms. And microorganisms literally means tiny living things. So all of the different tiny living things that are qualified as microbes are as follows. We've got bacteria. Bacteria are single-celled organisms. And there's lots of different types of bacteria. We'll spend a lot of time on those in this class. There are archaea. Archaea are a more ancient type of cell and really a different type of cell than bacteria and eukaryotes, which is what animals are made up of. They are not common pathogens, so we won't spend that much time talking about archaea in this class since it's more clinically based. But if you were taking a more traditional cell microbiology course, you would certainly want to learn more about them because they're pretty cool. They tend to live in really extreme conditions that resemble primordial Earth. And so they kind of have some really cool, extreme abilities. Protozoa are also single-celled organisms, and they are of the eukaryotic cell variety. And they are also pretty neat. There are some that are pathogens that we'll talk about, but there's far more that are not pathogenic or not human pathogens and just live in the environment. So we will spend a little time on some pathogenic disease-causing protozoa. There are fungi. Now, fungi can be microbes. They can be small, like yeast and different types of molds, or they can be macroscopic, like mushrooms. So the fungi is sort of a wide class. Helminths are kind of interesting. Helminths and viruses are kind of interesting exceptions to this list. So helminths are tiny. Some of them are tiny. Some of them are microscopic, but some of them are very large, like tapeworms can be several feet long. But tapeworm eggs are technically microscopic. So since a stage of the life cycle, particularly an infectious stage of the life cycle, is microscopic, we still categorize them in here with microbes, but just know some helminths are not actually microscopic. And then there are viruses. Now, viruses are, again, a sort of exception to this term microorganism because technically viruses are not organisms. They are not living things. They do not meet the definition of all the criteria of life. They cannot reproduce on their own. They don't actually have the genes or coding for proteins to sustain life on their own. So while they can survive and stay viable on surfaces for X amount of time, they cannot actually live and reproduce on surfaces that are not, they're not inside of a cell. Another thing, the fill in the blank here, that are considered microbes, but we really won't touch on in this class, are algae. And algae are they're photosynthesizing cells, organisms that are very small. They're almost like microscopic plants, and they really don't cause disease, so we won't discuss them in this class, but technically they are microbes. So this class will not go over all microbes in existence. This is a neat visualization of some of these different types of microbes, their size relative to things that we are sort of familiar with. So I'm just going to, this video is playing, so it's showing you 12-point font. Here's a coffee bean and a grain of rice and a sesame seed, so we know how small those things are. A grain of salt is practically microscopic. It's sort of like the limit of what you can see. Amoeba is a very large microbe. It's a little bit smaller than a grain of salt. Paramecium, these two are protozoa, and they are really fairly large cells. Human egg cells are also very large, much larger than a human sperm cell. None of these you can see with the naked eye. Here's a red blood cell, which is a very small human cell, which is still much larger than a yeast cell, much larger than an E. coli cell. An E. coli cell is about the same size as a mitochondrion, which is an organelle inside a eukaryotic cell like ours. And now we're getting down to some viruses down here, which are even smaller, much, much smaller than the bacteria, which are already tiny. And viruses actually have quite a range of sizes. So the rhinovirus is very, very small, about the size of a protein in our cells called a ribosome. Antibodies, which are a feature of the immune system, which bind to viruses and bacteria, are about this size. And now we're zooming into individual molecules of phospholipids or methionine, which is an individual amino acid, all the way down to a single carbon atom. And now we're going to zoom back out and see it in reverse. So here's the carbon atom, super, super, super tiny, much tinier than an antibody, which is a small protein. All right, these antibodies can bind to things like viruses, which could be as small as a rhinovirus or as large as the measles virus. All right, virus is way tinier than bacteria. Bacteria about the size of organelles in human cells. Bacteria is even smaller than a human chromosome. All right, then yeast is a little bit bigger, red blood cell, small human cell versus the egg cell, which is a large human cell, which is comparable with some of the protozoans, which are all smaller than a grain of salt. Okay, so just I'm a very visual person, and it's hard for me to visualize teeny tiny things that I have to just kind of imagine in my head. In my head, if something is tiny like a virus and a red blood cell, they kind of seem like the same size because they're both microscopic, but in reality they're actually very different sizes relative to each other. So some other things, sort of basic introductory concepts to understand that all of the science we're going to talk about in this course is evidence-based. And so that means that it was learned through a process of discovery known as the scientific method, where you make an observation, that's a weird phenomenon, I wonder why that happens, and you pose a question. You come up with a hypothesis or a sort of idea of what you think is going on, and then you test that hypothesis using experiments, well-controlled experiments. You conduct the experiments, you get results, you analyze the results, and if the results support your hypothesis, you continue to test it, so you repeat it. A key feature of science is repetition. The results have to be repeatable. You have to show that every time you do that experiment, you get that result, not that it wasn't a fluke result. If your results don't match your hypothesis, well, then you revise your hypothesis, come up with a new idea and a new guess, and test that. If you have repeated tests that support your hypothesis, that is evidence that supports your that you can create a scientific theory. So scientific theory is very different than how we use the term theory colloquially in the English language. So a lot of times when we use the word theory, what we're really talking about is a hypothesis, an idea, an estimated, guess. So when we say like, oh, I can't find my wallet, but I have a theory about where it is. All right, that's not a theory. That's a hypothesis. You have a guess. A theory in science is something that has been well tested, well documented, well evidenced, and is supported by a large bank of data. So it can be kind of confusing in like the lay media, because a lot of times people misconstrue when somebody says this scientific theory of blank. They're like, oh, well, it's just a theory. That's just an idea. But it's not. A scientific theory is not just an idea. It is actually an accumulation of evidence over many, many studies. So the theory of evolution is not an idea. It is actually scientifically proven through multiple experiments that show how organisms adapt, how their DNA changes, and how organisms in a population adapt to the environment that they are in. So how did microbes get here? So microbes were the original living things. They are the first life on planet Earth, and our best calculations and evidence that we have dates them to 3.5 billion years ago that they started. So life has been evolving for 3.5 billion years. So the three different types of cells that all things that are living are made up of are bacteria, archaea, and eukaryotes. So all three of these different types of cells originated, or thought to have originated, from the same ancestral cell that long ago divided and sort of gave root to these three different paths of its family tree. So bacteria and archaea are both types of prokaryotes, meaning they do not have a nucleus. So karyo means nucleus, pro means before. So these are the cells that existed before there was such a thing as a nucleus, right? Eukaryotes evolved to have a nucleus. And so eukaryote means a good or true or normal nucleus. Microbes are everywhere, everywhere. Sometimes taking this class makes you a little bit squeamish and a little too hyper aware of the microbes in the world around you. I hope not to give you anxiety about how germy this world is, because a lot of the bacteria or microbes on the planet are actually for good. We really couldn't live without them. And so it shouldn't be a scary thing. I hope that by the end of the course you'll find it a comforting thing that microbes are in fact ubiquitous. And this is a scientific term that you should know, vocab term. Ubiquitous just means they are found everywhere. Okay, so even in some of the most unlikely places, you will find microbes on planet Earth. They are found in Antarctica. They are found in boiling hot deep sea vents. They are found in super salty or super acidic environments. Your stomach, super acidic pH, but there are still microbes that live happily there. So as far as we know, there's really not a totally sterile environment on planet Earth. All right, microbes, I said the theory of evolution is an actual thing that can be demonstrated scientifically, experimentally. And a classic form of evolution that we see with microbes is, and that we can do in the lab, is show how they evolve to environmental changes like antibiotics. So all organisms change over time and adapt to their environment. And what evolution is it's just a series of genetic changes that get selected for and therefore become more common in populations. And where it's particularly relevant health-wise with microbes is antibiotic resistance. So this is how antibiotic-resistant microbes evolve. So let's say you have a large population of some bacteria. One of them, just randomly when it's being formed, it undergoes a mutation, a genetic mutation that causes it to be resistant to antibiotics. This is kind of an irrelevant mutation if there are no antibiotics around, and if there are no antibiotics around, that mutant might die off and its genes might die off with it. But if antibiotics are present, if you expose the bacteria to antibiotics, right, it kills off most of the non-resistant ones, and the one that is resistant survives and has more babies whenever it reproduces. And so more and more of the population becomes overcome with bacteria with that selective mutation, that adaptive mutation, and eventually all the ones that are not resistant will die off, and you end up with a population of solely resistant, antibiotic-resistant bacteria. So this one rare mutation becomes the norm when it's selected for, when there's that selective pressure of adding antibiotics to the environment. So you can take a very rare occurrence and make it the normal occurrence by adjusting the environment. And this is what happens with microbes, and this is how they evolve. The ones that survive best go on to reproduce, and since bacteria and viruses and other microbes tend to reproduce very quickly, this evolution can be observed in real time. We can't observe human evolution in real time because only like 3 or 4 generations, maybe 4 or 5 generations of humans are on planet Earth at any one, through one person's lifetime. Whereas bacteria can go through 3 or 4 generations in a couple of hours in a petri dish. So they just evolve much more quickly because they reproduce much more quickly. Microbes, while we will talk about all of the diseases they cause, they're actually really important for planet Earth. And some of the things that they do for us and for planet Earth is they are the main source of oxygen on this planet. So photosynthesis is really the main way that oxygen is generated for all of us oxygen-breathing animals and organisms. And, you know, in grade school we learn about plants and how plants do photosynthesis, and they're really important, and so trees are important. But it's actually microbes that are responsible for 70% of the photosynthesis on planet Earth. Algae in the oceans and cyanobacteria are actually produce much more oxygen than land-based plants. 70% of the planet is water, is oceans. So those water-based photosynthesizing organisms are incredibly important. They're important for climate control as well. A lot of other gases, not just oxygen, are produced by microbes and help to regulate climate, greenhouse gases, and regulate the temperature of the planet. Other things that microbes do, they can form associations with things like plants in order to help them get nutrients. And even our own bodies are colonized by microbes that help contribute to our health. So microbes really work in collaboration with other organisms to help them function optimally and to get nutrients. Other ways that humans have used microbes to our benefit, the most ancient way that we've done that is through food. We use microbes to make different types of food. So we use yeast in breads to make them rise and give them certain flavors like sourdough. A really important food preservation technique is fermentation. So alcohol is a way to actually preserve grains or fruit or vegetables that would normally spoil. So grapes don't last very long, they will spoil, but by turning it into wine, you can extract that, you can have a longer shelf life. Basically, you give the grapes a longer shelf life. So fermentation has been a way of producing sustenance that lasts longer. It also has the benefit or the fringe perk of, you know, making you feel good, a little drunkenness. So humans have also used it for that as well. And then there are a lot of bacteria that are used to ferment cheese or ferment milk. They produce acid that causes it to curdle and form cheese. So milk also is a food that will spoil and go bad and grow lots of undesired bacteria. But using specific bacteria, you can form yogurt and cheese, and those have a longer shelf life and also reduce the amount of lactose in the milk, which is not great for human guts. So we've used microbes to make lots of different types of foods, and we've been doing that for thousands of years. Much more recently, we have learned to befriend bacteria and microbes to do biotechnology. to manipulate these cells in petri dishes and use them as basically little microbial factories to produce proteins or drugs that are beneficial to us. A lot of antibiotics actually come from fungi. So we just basically grow a bunch of these fungi that naturally produce these substances, and we purify the substances. We also can genetically engineer organisms to produce substances for our benefit. So insulin is a great example. Insulin is a life-saving drug for people with diabetes, especially type 1 diabetes. 100 years ago, people with type 1 diabetes, if that was a death sentence. They were going to die from that disease, usually at a young age. Then scientists discovered the cause of type 1 diabetes, a lack of insulin production, and they managed to extract insulin from pigs, I think was the animal that they used, and they could purify that insulin and give it as a drug, and it was the first life-saving drug for diabetes. But it was crude because it wasn't human insulin, it was pig insulin, and it was expensive and pricey. You had to basically kill pigs to extract it, like grind up their pancreas. And when we learned how to do genetic engineering and we could sequence the human gene for insulin, we could insert it into bacteria and have the bacteria basically just pump out artificial human insulin. And we could purify that. So it was a much more pure, much more specific protein. And we were using bacteria to produce it rather than having to kill pigs. So it also made it a lot cheaper because bacteria can produce it very quickly. So it made drugs more accessible, less expensive, easier to produce, and more actually closely specific to human proteins. So it's been a real boon, but microbes have been really helpful in pharmacology and technology. We're also more and more using microbes to help us clean up our messes, not just cure us from disease, but clean up our messes. Microbes can evolve, and old dogs learning new tricks doesn't really apply to microbes. So new environmental pressures can result in adaptations, new adaptations of bacteria. Plastic didn't exist 100 years ago, but when we created plastics and started polluting the environment, some microbes actually evolved and adapted to be able to break down plastic. And so bioremediation is a way of using microbes to actually clean up pollution. There are also oil-eating bacteria that can help to clean up oil spills. And so we are trying to find ways to use these microbes and their natural talents and abilities to help clean up pollution. Most of what this class will focus on, though, there are all these good things that microbes can do, but what this class is mostly going to focus on are the diseases they cause. So microbes that cause disease are called pathogens. Literally, it's a word that means disease-causing agent. And so we'll talk about infectious disease. These are microbes that can, anything that can infect you, colonize and cause disease is an infectious disease. Some of these that we'll talk about are emerging infectious diseases. So there are new diseases that are cropping up, like coronavirus, for example, the SARS-CoV-2 virus. Coronaviruses as a family are an old family of viruses, but the new strain that causes COVID-19 is an emerging infectious disease. HIV is emerged in the 1980s, which is still relatively recent in human history, in our history of people living on Earth, but certainly not new in many of your lifetimes. Some infectious diseases like smallpox are ancient. Measles, malaria, flu, have been around for centuries, for all of recorded history. Re-emerging infectious diseases are ones that have been mostly controlled, but then come back after a period of being well controlled. These are just some terms in infectious disease. There are also a really interesting area of microbiology research is diseases that were not obviously caused by infection. So like chicken pox, measles, those are obvious diseases that are 100% solely caused by pathogens. But there are a number of other diseases, of more organic diseases in humans, that arise when human cells go awry, that we are starting to learn might be triggered by infections from pathogens. So, for example, certain cancers. Cancer is really at the heart, it's a genetic disease. It's a disease that's caused by genetic mutations within a cell that causes them to proliferate uncontrollably. Well, there are some viruses that can change your cellular DNA and lead to cancer. Two very classic ones are hepatitis C, which causes a lot of inflammation in the liver and can lead to liver cancer, and HPV, human papillomavirus, which is like almost the sole cause of cervical cancer, but it can also cause oral cancer, anal cancer, throat cancer, a number of other cancers. Gastric ulcers was a disease that in the 1950s, they had no idea what caused it. In fact, it wasn't until the 1980s that it was discovered that gastric ulcers are actually caused or triggered by the presence of a bacteria, Helicobacter pylori, and by treating the bacterial infection, you can treat the ulcers. Other diseases that have microbial candidates as triggers include type 1 diabetes. Obesity is more and more thought to be due to the types of microbes that you host in your gut rather than your diet, exercise, etc., even genetics. Schizophrenia is a neurological condition that may be triggered by infection. Coronary disease, multiple sclerosis, a lot of autoimmune conditions are actually starting to be linked to infection with certain viruses early in life that might trigger those conditions. So that is a really interesting forthcoming area of research, the role of microbes in non-infectious disease. So humans that are most susceptible to microbes and to pathogens are those who are immunocompromised. And there are more and more immunocompromised people on the planet as we improve healthcare. It sounds kind of ironic, but cancer, for example, a lot of cancer therapies involve drugs that destroy the immune system and leave people immunocompromised. So people are living longer from cancer. They're surviving cancer more, but that survival also makes them more prone to infection. As we age, naturally, our immune system starts to fail just like all of our other body systems, and we are living longer. And so we're living longer, but not necessarily healthier. So as we age, we have record numbers of elderly populations who are immunocompromised and who are more susceptible to infection, which is something we see with SARS-CoV-2 and the coronavirus pandemic. Another thing that is making us more susceptible to microbe infections is the evolution of drug resistance. So in the past century, we have developed a number of antibiotics which have been life-saving and revolutionary and revolutionized the treatment of microbial infections, but we have now entered the era of microbial resistance. We have overused those antibiotics, and now the bugs are smart, and these antibiotics are starting to not work anymore, and that is a huge problem. My favorite microbiologist is Louis Pasteur, and we'll talk about him more throughout the course, but this quote that he had I like. It says, "Gentlemen, it is the microbes who will have the last word." And I think what has always fascinated me about microbes and microbiology is that they are these tiny simple little organisms. They're microscopic, yet they can take down massive populations of organisms bigger than themselves. Classic example would be SARS-CoV-2, the coronavirus that causes COVID-19, killing hundreds of thousands of humans, which are more advanced creatures, I suppose, more advanced organisms than themselves. This tiny little virus can wipe out just these advanced organisms so easily. It's crazy to me. I have mad respect for microbes. So a little bit of history of microbiology, because I think the history of science actually makes it kind of more interesting. So if we go back in time, not that far, I mean back to the 1600s, all right, we didn't have microscopes back then. And so diseases that were caused by microbes, the existence of microbes was really unimaginable for people. They really thought a lot of disease was caused by magic, by spirits or demons or gods. And this concept of microbes was just beyond anyone's imagination. So the first window of discovery was in the late 1600s when the microscope was invented. So the first microscope was credited to Robert Hooke, who invented a microscope in the 1660s. And he liked to study plant material. He studied a lot of materials. And in studying a piece of cork, which is a type of tree, that you make wine corks out of, all right, he saw that when he looked at it under the microscope, it was divided into these little square shapes, all right, that reminded him of cells of living quarters in monasteries or prisons. So he called them cells because that's what the rooms of the monasteries and the prisons were called cells. He said it looked kind of like cells, like prison cells. So that's where the term cell actually came from. He was the one who coined the term cell for an individual unit of a living thing. About 10 years later, this linen smith, all right, Antony van Leeuwenhoek, he was a tradesman. He dealt with linens and garments, and but he liked grinding lenses as a hobby. And so he ground lenses to make these little microscopes, these very early microscopes. This is one of his microscopes. So this needle here, you'd put a little drop of fluid or something here. That's where your sample went. And this tiny hole here, that's actually where the lens is. So it's this tiny little lens. And you hold the thing up to your eye and you look through it and you can see things. And so he did that, and he just thought it was fantastic. And he would see these little things moving in a drop of water. So he would take scrapings from his teeth. He would take samples of rainwater that he caught in the drain, and pond water, and he'd look at it, and he saw these little tiny things moving around, swimming around, and he called them animalcules, which I just think is really cute. And this is a picture of some of the animalcules that he observed. And he documented everything. He took very careful notes, and he would write into these scientific societies, and they were like, you don't know what you're talking about. And they really disregarded him. And it wasn't until years later that his discoveries were actually appreciated and credit was given where credit is due. So he is largely considered the father of microbiology, even though in his lifetime he wasn't really taken seriously. Later on, people realized, oh, he was actually really onto something. And so it really was not appreciated in the 1600s. Most of the research on microbes happened more about 200 years later in the 1800s. But one other important experiment that I want to talk about in the 1600s was done by Francisco Redi. So in the 1600s, the idea of microbes and things evolving from invisible stages of life was very radical and not popularly believed. People thought disease came from magic, from gods. They also thought life in general just was created spontaneously by some divine being or magical spirit. And so they had this theory of spontaneous generation, that living things just spontaneously appeared, and animals just spontaneously got pregnant. And so this theory was called spontaneous generation or abiogenesis. Living things can come from non-living things spontaneously. Some of the theories they thought that rats came from piles of garbage. Large piles of garbage just spontaneously would cause rats to appear. And that rotting food spontaneously gave rise to flies. All right, to maggots and flies. And so Francisco Redi was like, no, this doesn't sit well. And he performed this very classic, very elegant and simple experiment where he took slabs of meat and he put one in a jar that he left open, and he put another in a jar that he covered with a piece of cheesecloth so nothing could get in. Okay, and he just left them both out on the counter and let them rot. All right, a week later, he finds that the piece of meat in the open jar is covered in maggots and there's flies all over it. All right, but in the covered jar, there are no maggots or flies inside the jar, but on top of the cheesecloth, there are maggots and there are flies. And so this showed that the maggots and flies come from the outside. They're not arising from the meat themselves, which is what people thought. They are actually coming from somewhere and gravitating towards the meat. He then took those maggots and put them in a jar and watched them develop into flies. So at that time, people didn't even realize that maggots were baby flies. They look very different. A lot of insects go through these metamorphoses where the infantile or larva stage looks very different than the adult stage. So until then, people thought maggots and flies were two different things. They didn't realize it was the larval form and the adult form. Okay, so that was one of the first classic experiments that supported this idea of biogenesis, that living things come from living things, that, the chicken comes from the egg, that inanimate objects do not sprout living things. Another classic experiment that was to support this idea of biogenesis, living things come from somewhere, come from something living. They don't spontaneously arise from non-living things, was Louis Pasteur. And he did these experiments called the swan neck flask experiment. So he took a flask with a long neck, like a bulb with a long neck, and he put some basically like chicken broth in it, some nutrient broth, and then he heated the tube so that he could curve it into this shape like a swan's neck. And he heated the broth, boiled it so that it was sterile, killed anything that was living in it. All right, and so this end was open to the air so oxygen could get in to support life, but it was curved in such a way that organisms could not easily float in here. Okay, so any dust or particulates that entered would get stuck in this little tube in this curve right here and not actually contaminate the broth. So it was a way to keep the broth open to air without allowing it to be contaminated by stuff in the air. And he kept this broth in this tube, in this flask, for a year, and it stayed clean and it stayed sterile and it was fine. And then he broke off that swan neck part so that now particulates from the air could drop directly into the broth, and within a day the broth became rancid and cloudy and clearly full of stinky, rotty growth. Okay, so he showed that there's something in the air, something invisible, and something particulate that can get into the broth and grow. It's not that there's something in the broth that arises spontaneously from the broth. It's something in the air that we can't see that causes this growth. And, we had microscopes, so he looked through microscopes and he started to identify these things. He also could look through the microscope and see there are no bacteria in this clear broth, but there were tons of bacteria, or microbes, I guess I should say, in the contaminated broth. They had grown and grown and grown. And so he theorized that this is what was happening in disease, a lot of disease cases too. So Louis Pasteur and Robert Koch were really the two fathers of germ theory. They were really important in establishing this germ theory of disease, that there are diseases that are caused by germs, by microbes, proliferating inside of an organism and making it sick. One of the diseases that they both studied, actually, was anthrax, where and Koch developed these really a sort of version of the scientific method that allows us to identify a disease that's caused by a germ very cleanly. So it was not until the late 1800s that we started to have that the reigning idea became that there are germs that cause disease. Things like smallpox that had been around for ages. People did not know that was caused by a germ. They thought it was caused by, like, sinning or breathing bad air. And now it started to come into clearer resolution that there are these little tiny microbes that can get inside you and proliferate and cause disease. So Louis Pasteur, the process of pasteurization is actually named after him. He saved the wine industry of France using this germ theory of disease. These vintners, these winemakers, would come to him with their spoiled wine and say, this wine is spoiled, it's sour, what happened to it? And he would look at it under the microscope and realize that there were certain bacteria that were causing it to become acidified or become sour. And he was able to sort of diagnose the problems of these wines. And the solution that he found was that they should boil the juice to heat it up before adding their yeast, and that would prevent the wine from spoiling. So this process became known as pasteurization. It's still used today for juices, for milk, for wine, for beer, where it's heated to kill any spoiling microbes. So Robert Koch and his postulates are really just a set of steps to identify that a pathogen causes disease. And he sort of famously came up with these by studying anthrax in sheep. And so he would find these sheep, they get sick, they die, cut them open, and he'd find that there were tons of these little bacteria in their bodies. And so he would take those bacteria and he could inject them into a mouse or a rabbit and find that they developed the same disease as the sheep, the same symptoms, and they also keeled over and died. And when he cut them open, he found the same bacteria in there. So it was a pretty clear cause and effect relationship. These little bacteria were causing these symptoms of disease and causing the death, and you could isolate those bacteria from all of the organisms that had that disease. So since people were starting to understand, okay, these tiny little organisms in the air and the environment can cause disease, maybe we should try to prevent that. And so aseptic technique was not a thing until germ theory existed. Doctors didn't wash their hands. They didn't know the value of washing their hands. And so it wasn't until the germ theory of disease that hand washing actually became a thing to promote good hygiene, and for doctors even. So there was a disease that was very common in pregnant women called puerperal fever. Basically, women after childbirth succumbing to infection and sepsis and dying from childbirth. And a lot of those infections, the infections were much more common in women who gave birth in hospitals versus with doctors versus those who gave birth at home with midwives. And it turned out it was because the doctors were coming from a surgery or from a morgue or from a patient who was sick with an infection to come catch this baby with dirty hands, no gloves, no hand washing, and they ended up transmitting infections to these women. And so aseptic technique became a thing. Joseph Lister was one of the pioneers in aseptic technique in the medical field. He first introduced sort of sterilizing, sterile surgery. So this is Joseph Lister, and he's using, I think, ethylene gas maybe to sterilize. This is the patient, and they've been covered with clean cloths, and only the area that's going to be cut open is exposed, and everyone has, it's very clean, washed hands, and it reduced the mortality from surgery by, I don't know what percent, but by a lot. So his surgeries were far less fatal than those of other doctors who were not using these sterile techniques because their patients were succumbing to infections as a result of their surgeries. Listerine, the antiseptic mouthwash, is actually named after Joseph Lister. All right, so those are some really big, important historical milestones in microbiology research. In the last century, in the 1900s and early 2000s, we have made huge leaps in the field of microbiology. We discovered what DNA was, the structure of DNA. We learned how to sequence DNA. We've learned how to manipulate DNA. And so a lot of discoveries in the 1900s led to Nobel Prizes in that area of being able to manipulate the genetic material of organisms, particularly of microbes, so that we could use them for things like biotechnology, like to make insulin, like I talked about earlier. So restriction enzymes, we discovered restriction enzymes, which are enzymes in bacteria that act as like molecular scissors, and they can cut DNA at specific sequences. So we can use those in a test tube to cut DNA at sequences so that we can cut and paste, literally cut and paste DNA. We learned about something called small RNAs. So these are tiny RNAs that can be used to turn genes on and off, to make them or not make them. There's a lot of different types of RNA that we'll talk about more in chapter 8 when we talk about genetics. That the type of RNA that we mostly think about is mRNA, messenger RNA, which also happens to be the type of RNA that is in the new vaccines for the coronavirus. But small RNAs have a totally different function and are really critical in controlling gene expression and also are a potential class of drugs, which companies like Moderna are studying. And then, of course, we have gained a new appreciation and understanding for the role of microbes, not just in disease, which is what we started studying in the 1860s, but in the early 2000s, we started to really appreciate the role of microbes in health. That having the right microbes in our microbiomes, our populations of microbes that exist within and on our bodies, and how they help to protect us and prevent disease and promote health. So that is an area of research that's very hot still in the 2010s and moving into the 2020s, how we can use microbes to help avoid disease and promote good health. So in order to understand microbiology and cell biology, we need to know some chemistry. Chemistry is sort of like the foundation of biology. So the important chemicals to know when studying biology are the 4 classes of what we call biomolecules. These are large groups of molecules that are really fundamental to life. And these 4 classes are carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates' main role is in energy, providing energy, and we can store carbohydrates as a form of energy storage. There are also some types of carbohydrates that we can use to build structural components of cells. So that's another function of carbohydrates. Lipids, primarily, again, for energy. They are a great source of energy And a really good way to store energy is as lipids. Lipids also make up our cell membranes, and there's also a number of hormones that are derived from lipids. So lipids have a lot of jobs. Proteins probably have the most jobs. Proteins, one of the most, they can provide energy, but there's so many other things they do that are really more important. The main thing is that they are enzymes. So enzymes are biological, they're catalysts, and they are made of protein, but their function is that they speed up chemical reactions. And life cannot take place without these catalysts. So enzymes have all kinds of functions. They speed up all kinds of chemical reactions, and they're very, very important. Proteins also help to make up our cell walls, along with carbohydrates and lipids. We'll find them in cell membranes. They're involved in transport across membranes. Proteins make up antibodies, which are important components of the immune systems. There's lots of different things. Proteins are super, super versatile and important. And then we have the nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And these are the molecules of inheritance. So this is how we store genetic information and express genetic information, or basically how we code proteins. Oh, we're getting an interruption. I'm on red. Sorry, recording a lecture. And I use like a red light, green light, yellow light system. So red means stop. Okay, so proteins are coded for by genes in our DNA, by the DNA, and the instructions to make those proteins are sort of in the form of RNA. So that's where proteins and nucleic acids come together. All right, so this is just a really nice visual chart that organizes information about these different biomolecules. So carbohydrates. Most of these biomolecules, by the way, are polymers, meaning they are long chains of repeating subunits. The subunits are called monomers. So carbohydrate monomers are called monosaccharides. Glucose is the most abundant and common monosaccharide of carbohydrates. We can link together these monosaccharides to form polysaccharides. So mono means one, poly means many. So these long chains of glucose form starch or cellulose. So there's lots of different structures we can form by linking these monomers together to form polymers. When it comes to nucleic acids and proteins, they are also polymers, long strands of repeating subunits. So the monomers of nucleic acids are called nucleotides, and we'll discuss those in more detail in Chapter 8. And we link those together to make long strands of DNA or RNA, which are polymers. Proteins are polymers of amino acids. The basic building block or monomer of proteins are amino acids. When we link them together in long chains, it's called a polypeptide, because these bonds that hold together amino acids are called peptide bonds. So polypeptide, many peptide bonds. Lipids are unique in that they are not polymers. So they are just individual units. So a very common type of lipid is called a triglyceride. It is a glycerol with three tails, three fatty acid tails. So triglyceride 3. So you really should know this slide here. You should know the 4 different categories of biomolecules. You should know what their different monomers are called. So, a nucleotide is the basic building block of what type of biomolecule? You should be able to say nucleic acid, right? There are a lot of things that are confusing in the terminology here, so this is where vocabulary really comes into play. For example, a lot of times people want to say an amino acid is a building block of a nucleic acid because they both have acid in them. Wrong. They might want to say that triglyceride sometimes sounds like glycerin or glucose or uh, glycogen. Those are all carbohydrates, but triglycerides are lipids. So that GLY prefix is often found in carbohydrates, but in this case it's not. It's found in a lipid. So there are some chemistry terms to sort of learn here and commit to memory, so you can remember which type of molecule is in which class, chemical class. So also, we'll talk a little bit more specifically about each of these classes. So let's start with carbohydrates. So carbohydrates, the simple ones, the monosaccharides, or when we combine two monosaccharides together, we get a disaccharide. These are what we call simple carbohydrates or sugars. So sugar that you eat, like when you get Dixie sugar crystals and you add them to your, cake batter, okay, or your coffee, those are actually disaccharides called sucrose. All right, so sugar is sweet tasting. A lot of monosaccharides and disaccharides register as sweet on our tongue receptors, and the purpose of them is to provide energy for our cells, primarily glucose. Glucose is a very important energy source for cells. So sugars, simple carbohydrates, their main job is energy. Polysaccharides, long chains of monosaccharides, usually glucose, can have two purposes. One is they can serve as energy storage spaces. So sort of like, I like to think of sugars as sort of like your checking account or the cash in your wallet, and polysaccharides as being like your savings account where, you put your money for long-term storage for when you need it later. So starch in plants is a form of stored energy, stored glucose. Glycogen is the form of stored energy in animal cells, like namely in our muscle cells and liver cells. Polysaccharides can also form very rigid types of structural materials. So cellulose in plant cells is what makes plants very fibrous and rigid, is the carbohydrate, the polysaccharide cellulose. One that's important in microbes is peptidoglycan. We'll talk about that in bacterial cell walls, the rigid structures surrounding bacteria. Peptidoglycan, the glycan part is the carbohydrate, the polysaccharide. The peptido part is peptide, so it's actually amino acids and carbohydrates kind of fused together. So it's part protein, part carbohydrate. So another job for carbohydrates is actually, as I like to think of them as like name tags on the surface of cells. So cells are often sort of decorated with these different patterns of carbohydrates on the surface. And those carbohydrates are these little sort of pink stringy things, pink ribbons here, okay? The green parts represent protein structures, proteins that are embedded in the cell membrane. And so these carbohydrate decorations, if you will, on the surface of the cell are actually code. They actually give information to other cells about where the cell is from. Is it from the liver, from the blood, from the brain? All right, different types of cells have different carbohydrate name tags, these different glycoproteins, these carbohydrate decorated proteins. They also can be kind of sticky. Think about like juice, which is full of sugar, and you spill it on the floor, and then it dries up and the floor is all sticky, right? So carbohydrates on the surface of cells also help them stick to things. So we'll talk about one of the cellular structures on the surface of the cell is the glycocalyx, which is basically like this sticky coating on the surface of bacterial cells that help them stick. Another important glycoprotein are the A and B and O antigens on the surface of blood cells. So you have type A or type B or type O blood. That's due to the carbohydrates that actually stick off the surface of your red blood cells. So those are the important features of carbohydrates. Now for lipids. Lipids are also important energy molecules. The most abundant type of lipid are triglycerides. And triglycerides contain a glycerol group and 3 fatty acids. And there's different types of fatty acids. Fatty acids can be categorized as saturated, which means they have all single carbon bonds, or they can be unsaturated, meaning they have at least one carbon-carbon double bond. If they have one carbon-carbon double bond, we say they are monounsaturated one, and if they have more than one, we say they are polyunsaturated. So poly meaning many. Lipids are stored in lipid storing tissues in animals. Adipose tissue is the lipid storage, the cells that their job is to store lipids for energy. When your cells need energy, there's plenty in storage. So it's like the ultimate savings account, I suppose. Another major class of lipids are phospholipids. So phospholipids actually look chemically very much like triglycerides. They have a glycerol group, and they have 2 fatty acid chains, but in place of the 3rd fatty acid chain, they have a phosphate group instead. And this phosphate group makes all the difference in the chemistry of this molecule. So lipids, by definition, are not water soluble, and triglycerides are definitely not water soluble. Think oils. Oil and water does not mix, okay? But phospholipids can actually mix with water, but they kind of do a weird thing when they're put in water. So the phosphate group is charged. It has a negative charge, and charged things get along well with water. Water is also partially charged. And so things with charges dissolve well. In water, we say they are hydrophilic. They like water. But things that contain lots of carbons and hydrogens are hydrophobic or nonpolar, and they do not mix well with water. And so what we have here is a molecule that is both polar and nonpolar. The head group likes water, but the tail group does not like water. So when you put phospholipids in water, one of 2 things will happen. They will either form something called a micelle, basically a sphere, where all of the those charged head groups, polar charged head groups, are on the outside of the sphere, and all those nonpolar, hydrophobic, water-fearing tails, fatty tails, are on the inside. Okay? And then this molecule is comfortable floating around in water because all of the exposed surface is those hydrophilic heads that like the water. Another structure so this structure is important because, and relevant, because the coronavirus vaccines that are out right now through Pfizer and Moderna, the mRNA vaccines, are packaged using basically a type of lipid that is structured very much like phospholipids, and they form these spheres, and the mRNA is carried on the inside of these spheres that form from these lipids, these lipid nanoparticles. Another structure that phospholipids form is something called the phospholipid bilayer. So when you put them in water, instead of forming a sphere, they basically form these 2 layers where the tails interact with each other and the head groups are exposed to the water on either side. So our cell membranes, all cell membranes actually, of all organisms, are basically formed primarily from phospholipids, a phospholipid bilayer. So you can see the phospholipid bilayer here. It also has all these other things embedded in it. These are mostly proteins that embed in it, and here's some glycoproteins with some carbohydrates sticking off of them. All right, but the cell membrane, the foundation of the cell membrane, is that phospholipid bilayer. Lipids are the main component of our cell membranes. All right, and then the third important class of lipids are the sterols. Cholesterol being sort of the poster child of this group. Cholesterol gets a bad reputation, heart disease and whatnot. It's actually an essential nutrient. It's an essential molecule for life. It's important for building cell membranes. Cholesterol is a component of cell membranes that adjusts the flexibility of a cell membrane. We also use cholesterol to make a number of hormones, all of the sex hormones, estrogen, testosterone, cortisol are all derived from cholesterol. And vitamin D is actually derived from cholesterol. So cholesterol and sterols are very important. They're still classified as lipids because they are not water soluble. They are non-polar molecules. Waxes are also a class of lipids, but I'm not going to get into those. Now onto proteins. So we've talked about carbohydrates, lipids, and now proteins. So proteins are made up of amino acids. Those are the building blocks. And we link them together in long chains that are called polypeptides. They're called polypeptides because the bonds that link them together are called peptide bonds. There's something like 20 different amino acids. There's 20 common amino acids. There's other amino acids too, but these are the basis of all proteins are these 20 amino acids, and you don't need to know them, but they're just listed here for reference. All right, and we can make really an infinite number of proteins simply by linking together different amino acids in different orders. So it's kind of amazing to think that there's really only 20 different amino acids, but there's like an infinite number of proteins we can make. And it's kind of like if you think about language and words. So the English language uses an alphabet of 26 letters, but there's millions of different words you could come up with simply by combining those letters, different, order, different lengths. And so through that same mechanism, we can make, millions of different proteins by combining amino acids in different orders and different lengths. So a protein is really just a long polypeptide. So a polypeptide that has 50 amino acids or more is generally how we define a protein. You can have shorter chains of amino acids, and we technically don't call those proteins because they're so small. But that's just some semantics really for you in biochemistry. So a peptide is what we call a short chain of amino acids, and a polypeptide a longer chain of amino acids, and a protein is really just a very long polypeptide. So proteins fold and form these specific three-dimensional shapes that are fundamental to their function. So we have different levels of protein structure. The simplest level is called the primary structure, and that is simply the sequence of amino acids. That is its primary structure. It's leucine, aspartate, alanine, valine, arginine, blah, blah, blah. The sequence of the amino acids. If the beads on a string, if you will. But those beads on a string can sort of coil up. I should have a pipe cleaner to be able to do this little demonstration for you, but I don't, sorry. So think of a pipe cleaner wrapped around my finger and it forms a coil, all right? We call that an alpha helix, right? And think of the other end of the pipe cleaner, I fold it back and forth like a fan. So I'm just folding it back and forth. That structure is called a beta pleated sheet. So in a long chain of amino acids, you might have multiple little sections that contain an alpha helix or a beta pleated sheet. Then if you take that imaginary pipe cleaner and you fold it all up into like a knot, okay, that is what we call the tertiary structure. So these individual segments may have an alpha helix or a beta pleated sheet, but the whole thing and its folded up structure in its entirety is called the tertiary structure of the protein. So the fully folded tertiary structure is the functional protein. If you unfold it, all right, and you take it down to its secondary or primary structure, it won't work. Whatever the job of that protein is, it's probably not going to work. Some proteins are large complex structures of multiple subunits. And so if a protein actually is made of multiple subunits, then we say that it has quaternary structure, a fourth level of structure, which is all those tertiary proteins coming together. Okay, so proteins have a variety of different functions. They can act as enzymes and catalyze reactions. They can form hormones that are basically cellular messengers that tell cells to do things. They're the components of antibodies, which are really important immune molecules. Proteins help us in fluid balance in our bodies. They help with acid-base balance. They can serve as buffers. They serve as channels, transport channels in cell membranes that allow things in and out of the cell. They can work together with lipids and form these spheres that help transport things through the body, and they form important structural components of things like muscle. Actin and myosin are the proteins that are involved in muscle contraction. They're responsible for muscle contraction. So many different things proteins can do. But they can't do those things if they're not in the correct structure. So the actin, for example, or antibodies in their three-dimensional structure have an important function. But if you were to unfold the protein and ruin its structure, so you're not breaking it in the sense that you're not breaking these peptide bonds, so the primary structure is still there, but if you destroy that tertiary structure, the three-dimensional structure, it loses its function. And so the process of taking a protein and unfolding it and destroying its tertiary structure is called denaturation. And that's just an important vocabulary term to know. So proteins can be denatured. Sometimes denaturing is permanent, and the protein can't refold. Sometimes it's temporary, and exposing it to different conditions will allow it to refold. All right. So one of the things, lots of different things can cause proteins to become denatured, changing their pH, changing the temperature. All right. One of the reasons that very high body temperature is dangerous is because high heat denatures proteins. It denatures important proteins for cells to function. So if your body becomes too hot, it starts to denature proteins and they start to lose their function. It's also why heat helps to kill bacteria and viruses, because they are also made of proteins, and if their proteins become denatured, then they cease to be able to function and live. And the last class of biomolecules that are important are the nucleic acids. We have DNA and we have RNA, and we'll get much more into these in Chapter 8. But some of the key differences and similarities between them. DNA is our genetic material. We pass on DNA from generation to generation, and it is double-stranded, and it lives in the nucleus of the cell, hence nucleic acid. RNA is a more unstable nucleic acid, and it's usually single-stranded, and we sort of make temporary copies of the DNA in the form of RNA, and then we destroy them. So I think of RNA as almost like photocopies of the DNA. But there's lots of different types of RNAs, so I'm really generalizing here. There's messenger RNA, transfer RNA, ribosomal RNA, MT and rRNAs that we'll talk about in Chapter 8. There's also really interesting small RNAs, interfering RNAs, that we won't get into in this course, but you would if you took like a genetics course. So DNA is found in the nucleus, RNA is made in the nucleus, but then it gets transported out of the nucleus into the cytoplasm. One is double-stranded, the other is usually single-stranded, and they are made up of 4 bases: adenine, guanine, and cytosine, and then the 4th base in DNA is thymine. The 4th base in RNA is uracil. So that's another difference between them. So one of the nucleotides that makes up RNA is adenosine monophosphate. You add 2 phosphates to that molecule. So here's adenosine monophosphate, which is a nucleotide building block of RNA. Okay, add 2 more phosphates to this molecule, and you have ATP, adenosine triphosphate, which is a really important molecule in cells. It is the energy currency of the cell. Cells use it for energy. So why is it the energy molecule of the cell? Well, it's because those 3 phosphate bonds have a lot of energy in them, particularly this last one. And when ATP reacts with water, it's a very exothermic reaction. It releases a ton of energy, and that energy can be harnessed by the cell to do work, like mechanical work, like muscles contracting, or chemistry work to basically fuel a chemical reaction that requires energy input. So ATP is a very important energy molecule in the cell, but it's technically a part of the nucleic acid family. It's really a modified nucleotide. So the last thing in chapter 1 talks a little bit about taxonomy, or how we classify living things. And you may have, recall from, or not recall from grade school learning about the different classification system of living things, all the way from domain, kingdom, phylum, class, order, family, genus, and species. This taxonomical system was developed by Linnaeus. It's the Linnaean classification system. And it allows us to basically organize living things into groups based on their relation to each other. So things that are the same species are the same organism. And then sort of the next level of classification is genus, family, order, so on and so forth. And so we can give each organism a name in the scientific system. We give them a Latin name. The sort of scientific term for naming things using a system of rules is called nomenclature. And the way that we name organisms is by naming their genus and their species. And there's a specific way that we write this. It's in italics, and the genus is capitalized and the species is lowercase. So we will classify, just like we classify macroorganisms, animals, insects, we also classify microorganisms, but it's a little bit different on the microorganism scale. So here's an example of using the Linnaean taxonomy system. So the domain would be, there's 3 domains: eukarya, archaea, and prokaryotes. So humans, for example, are in the domain eukarya, but so are paramecium, which are protists. Okay, so a lot of different organisms fall in the same domain. And then as we move down that taxonomy system, we get more and more specific. I'm not a huge fan of taxonomies. I'm not going to focus much on this in this course. It's just how things are named, and it's also a way of sort of building family trees and seeing how things evolved and how they're evolutionarily related to each other through shared traits. And so the main thing I want you to take away from this is that when we are writing the names of microbes, the scientific names of them, it is important to write them correctly. They should be written, punctuated correctly, or formatted correctly so E. coli, the full name of E. coli is Escherichia coli. These are the incorrect ways to write it. The correct way to write E. coli is in italics. The E is capitalized in Escherichia, and the C in coli is lowercase. So that's the end of chapter 1. I do have some questions from the publisher that you can go through on your own. I don't find them all that helpful, and I might ask them in live lecture. If I was live, I would throw these questions at you, but I'm going to stop talking now. The end.